Okay, let's go ahead and get started. Welcome to Medical Grand Rounds. It's really such a timely topic for us to discuss, especially now given that we're at the beginning of this second surge of patients and of people testing positive for SARS-CoV-2. We have two guest speakers from the Rockefeller University that will focus on neutralizing antibody response to SARS-CoV-2 infection. I'll be moderating the question and answer period at the end of the formal presentation. So please put your questions in the Q&A box and we'll be tracking them and trying to get to all the questions. Let me introduce our two speakers. Our first speaker is Theodora Hatziioannou. Dr. Hatziioannou is a research associate professor in the Laboratory of Retrovirology at the Rockefeller University. Our second speaker is Paul Bieniasz. Dr. Bieniasz is professor and director of the Laboratory of Retrovirology at the Rockefeller University and a Howard Hughes investigator at the Rockefeller University. Both have been exceptionally creative and productive investigators that have focused previously on HIV replication and host defense. When SARS-CoV-2 hit, they quickly pivoted and began to study that organism and the response to that virus. And they've done incredibly important work in neutralizing antibody and the development of therapeutics that will potentially impact the course of COVID-19. So it's a pleasure to welcome them, and I really look forward to their presentation.
Well, thank you very much, David, for the introduction, and thank you, Eric Poschler, for the invitation. That was months ago. And in case you were wondering why we're not keeping social distances here, is because we are actually also husband and wife, so we try not to. So I will give the first part of the talk after a small commercial break. To promote the Principles of Virology 5th edition. It's a textbook about all viruses that basically tries to introduce them based on principles rather than individual viruses. And I was very fortunate to work with the really wonderful and very esteemed team of colleagues to bring this book into an updated edition that's going to be released in print very soon. It's already available online. So, and I was also extremely happy to contribute to the covers, which now include my two favorite viruses. As David mentioned, we used to work, or we still are working on HIV-1, everything from innate immunity, interaction with the host, animal models. But when the SARS-CoV-2 pandemic spread into the US and particularly into New York, where we are, we thought very hard about it and decided that we had something to contribute towards this pandemic also. So a large part of our effort now focuses on this new virus, and in particular its interaction with the immune system and the antibody response in people infected against this virus, which is going to be the major topic of our talk. So just a very brief introduction. I'm sure you're all very familiar with the number of the infected cases in the world and in US in particular, and the second or third wave that is currently going on. So I won't reiterate the numbers. I will just start with our work. So this is a very well-known electron micrograph of the virus, and you can see the spikes decorating the surface, which are shown here. And this is the just the schematic representation of the spike glycoprotein, or the envelope glycoprotein that decorates the outside of the virions. And it has two parts, the S1 and the S2, various protease cleavage sites, which we are not going to really dwell on. And the extracellular part of this protein has been crystallized, and it's a trimer. and contains the receptor binding domain up here. And of the 3 trimers, in 2 of them, the RBD is in what is called the down conformation, and only 1 out of the 3 is in the up conformation and available to engage the cellular receptor, which is ACE2. So when we first were thinking about what we could do, we have, as I mentioned, experience with HIV and VSV. So what we did was to take the spike glycoprotein and use it to pseudotype HIV-1 or VSV particles, and that is to produce particles that the core is either based on HIV-1 or VSV, and that carry the nanolook reporter gene, and just supply this spike protein in trans, the protein gets incorporated, and we can also use these viruses to infect cells that we have engineered to overexpress ACE2, and we get some really nice dynamic range that can be read very easily and is adapted to a 96-well format. So we can perform a large number of assays, that is for a lab, with this system. And what we mainly have used this assay for is to measure the neutralizing responses in plasma from convalescent patients. So you can take plasma or sera from these patients that presumably contains antibodies against SARS-CoV-2 and incubate it with our viruses, and then measure how well this plasma inhibits the interaction of the spike with ACE2 and inhibits infection of these 293 T cells. And as you can see here, we use plasma dilutions to incubate with the viruses, and you get a range of activity. So the greater the amount of antibodies that can neutralize the virus in the plasma of these patients, the lower the infectivity. And we get similar results using either VSV or HIV. And what we have used as a measurement for this activity is what we call NT50, which is the concentration or dilution of plasma at which the maximum infectivity is decreased to 50%. So, of course, it was important to validate that our assay really represents neutralization of the natural SARS-CoV-2 variant, and we did that in collaboration with Charlie Rice, also here at Rockefeller, where he took a same set of plasmas and measured their neutralization of SARS-CoV-2. And as you can see, both our assays correlate quite nicely with neutralization of the bona fide virus. So we could use this assay to probe the responses in patients. And we have done this now for almost 450 patients from various cohorts, some from a Rockefeller University cohort, the New York Blood Center cohort, and a cohort from Scotland, the NHS. And this is the distribution of NT50s in the plasma samples that we have analyzed. So 20% of the samples that we see have negligible neutralizing activity, at least below the limits of our detection system. And only about 10% are what we call elite neutralizers that have high neutralizing activity in their plasma. This is above 5,000. The majority of samples, as you can see, have very low to modest activity against SARS-CoV-2. And I think this is in agreement with a lot of data presented by a number of labs around the world. So some of the factors that influence levels of SARS-CoV-2 antibodies, as I'm sure you're well aware, males tend to have higher levels of neutralizing antibodies than females. They also tend to get more sick. So it's possible that this correlation is due to higher levels of virus replication in their body. And also, again, as noted by many other studies, patients that have severe cases tend to have higher levels of neutralizing antibodies than cases with more mild symptoms. And I have to say that the majority of the patients for which I will show you data in the subsequent slides are actually mild or cases. So one question that a lot of our colleagues and ourselves were asking is all these assays that measure SARS-CoV-2 antibodies, measure are actually based on just detection of antibodies and do not determine whether those antibodies can neutralize SARS-CoV-2. So are they any good at predicting whether the antibodies are neutralizing or not? And we did this again in collaboration with the New York Blood Center here in New York and the NHS in Scotland. And you looked at a number of patients whose antibodies were assayed with these various assays. Some are particular to the UK, others are used here too, like the Abbott, the Ortho, and correlated the values obtained using these serological assays with the neutralization assays that we performed. And as you can see, some of them correlate quite nicely, whereas others really don't. But even, so here, of course, the axis of the measurements for each assay differs. It depends on how the assay units are defined by the manufacturer and varies greatly. So one thing I would draw to your attention to is the Ortho Igg assay, which is currently used by the FDA to determine which donors are going to donate plasma for therapy or for convalescent plasma therapy. So this is the cutoff that they're using. But as you can see, when you look at the values of these across the neutralization efficiency of this plasma, you have a really wide range. So all these plasmas are going to be selected for use. But as you can see, the amount of neutralizing antibodies within these plasma samples varies greatly. And personally, I don't think I would be so happy if I was sick to receive plasmas that score at this level of neutralization antibodies. I would much rather prefer higher values. And again, all this data I'm showing you here were performed with plasma obtained shortly after infection, within 4 to 5 weeks. So what happens, and this has been in the news a lot, particularly this week again, actually, happens over time. How do these what happens to the antibody levels several weeks post-infection? And this is an assay that we performed with the NHS cohort in Scotland and measured at 3 to 4 different visits post infection. So the first visit is around 4 weeks, 8 weeks, 12 weeks, and then later on. And with patients measured with 4 different assays. And as you can see, for the majority of these serological assays, the level of antibodies stays steady. The only one that declines is the Abbott assay. Now we can look at this in a bit more detail over, as I said, visit 1, 4, 8, and 12 weeks post-infection. And again, this diazorin and Siemens assay appear to remain stable over time, and they measure either the spike protein or are specifically directed against the RBD, whereas the Abbott and Roche assay that measure the N protein, the nucleocapsid protein of the virus, the Abbott declines, but the Roche doesn't. So this decline for the Abbott test does not appear to be dependent on what viral antigen the assay detects, but it rather seems to be something specifically associated with this assay. But we know, so it appears that at least antibody levels as measured by these assays don't decline over time. But what happens to neutralizing antibodies? Well, this is where our assay came in, and we measured all these patients from visit 1, 2, and 3, color-coded here. And it's really very clear that antibody levels, neutralizing antibody levels, in contrast to what is detected with most serological assays, drop over time. And the higher the levels, the more significant the drop. But we hardly had any patients that did not decline. So if you are looking at the neutralizing antibody levels, it's very clear that within 12 weeks, they declined by about 50%. And this decline is not reflected in the antibodies measured by these high throughput serological assays, even when they measure the same antigen that is targeted in our assays. So in short, these assays cannot be used really as a predictor for neutralizing activity in in convalescent patients. They do appear, however, to be optimized for sensitivity. So even very low levels of antibody are readily detected with these assays. So it really depends what you're trying to do when you're using assays like this. But as I mentioned before, you cannot predict neutralizing antibody levels. Another question that's been asked recently is whether pre-infection with seasonal coronaviruses affords any protections against SARS-CoV-2, and in particular, whether any of the antibodies or neutralizing antibodies raised against seasonal coronavirus have any cross-reactivity against SARS-CoV-2. So again, we did this with the NHS cohort, where they had plasma samples stored that were obtained prior to the SARS-CoV-2 pandemic, and that had been confirmed by PCR to be positive for any one of these 3 seasonal coronaviruses. And we tested the neutralization activity against all these seasonal coronaviruses, but I'm only showing you one, this OC43 in particular. And as you can see, they do neutralize OC43 very efficiently. And they also some of them show some neutralization against the other viruses. At the moment, we don't know why. The most probable explanation is that these individuals were also exposed to these other seasonal coronaviruses. In sharp contrast, they show no cross-neutralization activity against SARS-CoV-2, whereas, as I showed you before, COVID-19 sera do neutralize SARS-CoV-2 very effectively. So in terms of neutralizing antibody, there doesn't appear to be any cross-reactivity between those raised against seasonal coronaviruses and SARS-CoV-2. So we were actually in collaboration with Michelle Nussensweig lab, who is an expert in cloning antibodies, interested in isolating human antibodies specifically against SARS-CoV-2. And for that, we used patients recruited at the Rockefeller cohort. And you see that they had a range of neutralizing activities. And Michelle used an RBD bait to select individual B-cell clones and isolate paired heavy and light chains from these cells from 6 different patients. So obviously we picked the 2 patients that showed the highest neutralizing activity, but also included 4 that had modest to very low neutralization activity. So these circles represent the number of antibodies isolated from each patient. This is the total number per patient. Lines in white indicate sequences that were only isolated once, and in gray, sequences that were isolated multiple times, probably representing clonal expansion of the corresponding B cell. And in red, these colors, or blue, are antibodies that were almost identical in sequence between different patients. So all these antibodies were tested for a binding to RBD by ELISAs, and then a few were selected for neutralization assays that we performed. And you can see that in each patient, we get a range, or rather in most patients, we get a range of neutralizing activities of individual antibodies. And the range of activity of individual antibodies doesn't always correlate with the overall neutralization activity found in the plasma. For example, here we have really potent antibodies that neutralize the virus very efficiently, and this plasma itself has a very high neutralization activity, but we get equivalently potent antibodies from patients that have very low neutralization activity overall. So this finding suggests that a lot of patients are capable of mounting an immune response that includes these really potent neutralizing antibodies. They're just probably not making enough of them to affect the overall neutralization activity of their plasma. So then the next part of the talk, I will focus on our 3 favorite antibodies, this C121, 135, and 144 that were isolated from these individuals respectively. And this is the neutralization activity against a pseudovirus. And they also were able to neutralize SARS-CoV-2 as, if not slightly more efficiently than our pseudoviruses. So the next thing we did was, in collaboration with the Sheehan lab, is to test the ability of these neutralizing antibodies to inhibit SARS-CoV-2 infection in a mouse model. And she points out that the SARS-CoV-2 spike is unable to use the mouse ACE2 receptor very efficiently. But the Sheehan lab adapted this spike and introduced 2 amino acid changes that now make it capable of using mouse ACE2 as efficiently as human ACE2. And we, of course, confirmed that all the antibodies that we were testing were able to inhibit this mutant spike as efficiently as the wild-type spike. So the experiment we performed was a prophylaxis experiment in mice where the antibody was delivered in mice IP that were 12 hours later challenged with SARS-CoV-2 intranasally. And 2 days later, the mice were sacrificed and virus titer were measured in the lungs. And this is the results with individual antibodies. This is what the titers we get in untreated animals. And you can see that each of the antibodies decreases the titers of the virus quite dramatically, some to undetectable. And then when you start using combinations of this antibody at these doses, you can get apparently sterilizing immunity where we cannot detect any virus in the treated animals. And these 2 antibodies are currently in clinical development, and we're hoping to eventually get them to the clinic. Now, Paul will take over.
Okay, so let me pick up from there. As Theodora mentioned, sort of one of the culminations of our work has been to identify these incredibly potent monoclonal antibodies that, as she mentioned, are now going forward in clinical development. Now, of course, whenever anyone brings a therapeutic to the clinic against a virus, one of the first questions one needs to ask is, how likely is it that resistance will emerge to that therapeutic? And that actually brings up a broader question of antibody immunity in SARS-CoV-2. So that obviously would have implications for monoclonal antibody therapeutics, but also for vaccines and for convalescent immunity. So how likely is it that antibody resistance will emerge in SARS-CoV-2? Well, there are a number of factors that could influence that, and there's no simple answer to that question. But if we consider a hypothetical transmission chain of individuals here, where a virus strain that's initially antibody susceptible in a particular individual becomes antibody resistant and then is transmitted in an antibody resistant form, the probability of that event occurring is affected by a number of factors. Okay, some of those factors are intrinsic to the virus. So replication fidelity, we know that SARS-CoV-2, like all coronaviruses, has a proofreading polymerase. So its tendency to generate sequence diversity is significantly less than most, if not all, other RNA viruses. However, the sort of antidote to replication fidelity in terms of generating genetic diversity is population size. And we know that the population sizes for SARS-CoV-2 are enormous. There are upwards of 10 to the 8, 10 to the 9 RNA molecules, even in a single swab taken from an individual. That basically means that every possible variant of SARS-CoV-2 that can be generated has been generated in some individual at some point during the epidemic. And it's likely, in fact, that a large fraction of the possible variants even exist within a single individual. Now, it's not only genetic diversity that influences the possibility of resistance emerging. Things like transmission dynamics will also determine the number of times that an antibody susceptible population will encounter antibodies, and that antibody would then be able to impart selection pressure on that virus. And if one takes this hypothetical scheme of what might happen in an infected individual, where virus load increases and then decreases, and presumably with it the probability of transmission, the degree to which the neutralizing antibody response overlaps with that transmissibility window again will impact the propensity of the virus to encounter and acquire resistance to antibodies. Now, that's, of course, before there has been any intervention or any second waves, reinfection, for example. As Theodora mentioned, immunity is likely to wane, and based on everything we know with other coronaviruses, reinfections are likely to occur, potentially in the context of sub-therapeutic or sub-protective levels of antibodies. We and others are planning to use monoclonal antibodies as therapy. Again, that's going to increase the number of encounters between virus and antibody. Convalescent plasma therapy has already been given to tens of thousands of individuals. Most, as Theodora mentioned, have received suboptimal levels of antibodies, again, increasing the number of encounters between virus and antibody. And of course, as vaccines roll out, it'll be critical to understand whether those vaccines induce antibodies that remain protective. So I would argue that given this scenario, it's for the first time we have a global pandemic where all of these therapeutic or protective modalities are being thrown against a globally prevalent virus. Somewhat difficult to predict whether antibody resistance will become a problem. So we don't know at this point, but what we can at least do is try to develop tools to get at that question. And this is a tool that we have developed that has enabled us to try to get at that question. So what this is a recombinant virus. It's based on vesicular stomatitis virus, a rhabdovirus distantly related to rabies. And what we did was to take away its envelope protein and replace it with the gene encoding the SARS-CoV-2 spike protein. And we're also including a GFP gene so we can track infected cells. So the reason we did that is this virus grows rapidly to high titers. And perhaps more importantly, it doesn't have a proofreading polymerase. So we can, in relatively small populations of virus that we can generate in the laboratory, cover a much greater proportion of the potential diversity in the SARS-CoV-2 spike protein. So we built that virus, we've adapted it in cell culture so that it grows very well indeed. When we adapted it in cell culture, it picked up a couple of mutations at the protease cleavage site, one that Theodora mentioned, a couple of other passenger mutations that we don't think are of any consequence, but just in case they are, we do all our selection experiments with 2 different strains. So we can infect cells, we can isolate individual plaques. This virus grows very rapidly to very high titers. And perhaps most importantly, of all the recombinant viruses we've made, this virus most accurately recapitulates the neutralizing properties of SARS-CoV-2 itself. So here's the IC50 for 20 monoclonal antibodies against the authentic virus and our hybrid virus that we have built. So here's the experiment that we've been doing. What we do is to take a large, diverse population of these viruses that have acquired mutations in spike. We take that population of viruses and then we neutralize it with high levels initially of monoclonal antibodies. Put that mixture onto cells, and then any viruses that grow out, we passage a couple of times and then sequence and try to ask what mutations has the antibody selected in that virus population. So here's some actual data. So this is one well of a 12-well plate where we've put 1 million infectious units of that GFP positive virus on, and so you can see all the cells turn green. But if we use one of our favorite antibodies, you can see that most of those infection events are blocked. Most, but not all. You'll see the odd green spot here. And if you look at particular places in the monolayer, you'll see where individual infection events have begun to spread. Here's one example, and here's another example. These are putatively resistant variants of the SARS-CoV-2 spike. And if we take supernatants from this culture, the virus indeed grows in the presence of antibody, and then we sequence the virus and see what has happened. So I'm going to show you a number of charts here where the SARS-CoV-2 spike sequence is represented on the x-axis as a codon position, and then on the y-axis is how frequently we find a variant at each one of those positions. And so when there's no antibody, there's very little variation. When we use one or of our favorite antibodies, it's clear that variants at particular positions in the spike protein have a selective growth advantage, and they crop up through this passaging procedure. So what we then do is isolate those individual viral variants. We pick the individual plaques, sequence them, and we find, for example, in this case, we've got a particular mutation in the spike protein. And unlike the wild type virus that's very nicely neutralized at picomolar concentrations of our favorite antibodies, this mutation is now completely resistant to these two antibodies, but retains full sensitivity to this antibody. Conversely, a different virus that was selected with this antibody is completely resistant to that antibody, but retains full sensitivity to the other two antibodies. So that enables us to construct sort of epitope maps of where these antibodies are imposing selective pressure. We've complemented that approach by looking at naturally occurring substitutions in the spike protein. You'll be aware that there now exist over 100,000 SARS-CoV-2 sequences in databases, and a huge number of individual variants are naturally circulating. So what we've done in this experiment is to make spike proteins that have mutations that are naturally occurring, and we pick out the mutations that are in the RBD region of the spike in the vicinity of where the assays that I've just told you about tell us approximately where those antibodies bind. And so now we're measuring the IC50s, the potencies of those antibodies against all of these naturally occurring spike variants. And you can see for C121 and C144, there's a cluster of mutations here that confer partial or complete resistance. Similarly here, whereas for C135, all of those mutants retain full sensitivity, but some mutations over here confer resistance. And we can map those resistance mutations onto a structure of the RBD spike. And this is, in fact, where on the RBDs these antibodies bind. Information like this can tell us what combinations of antibodies should be included in potential therapeutic cocktails. Now, just a quick word about how prevalent these pre-existing resistance mutations are in naturally circulating populations. Again, this is the sequence of the SARS-CoV-2 spike represented on the x-axis. And this is how frequently variants at each position are found in the sequence database as it currently exists. I'm sure many of you have heard of this rather famous mutation, but the ones I direct your attention to are depicted in the colored triangles here. These are variants that we know confer resistance to antibodies that are studied by us in red and in green, or by the Regeneron group in the red and the blue. Some of the resistance mutations are shared by the antibodies we found and the ones found by the Regeneron group. Now, the actual position on the y-axis we shouldn't take too seriously, because this sequence database is heavily influenced by founder effects, the proximity of labs to outbreaks that contribute sequences to the database. But the point I would make is that the most prevalent resistance mutation is still present at less than 1% of circulating viruses. So these resistance mutations are there, but they're there at very low prevalence currently. Okay. So obviously this type of data inspires us to make antibody cocktails. And so here's essentially the same experiment that I've just shown you, but instead of using 10 micrograms of a single antibody, we split that dose in half between two antibodies that have non-overlapping profiles of resistance mutations. And while you still get the odd infected cell, if you zoom in, what you'll see is that those infections, individual infection events, aren't expanding. Okay, these are antibody susceptible variants that we just haven't added enough antibody to neutralize every infection. And if we do essentially a related experiment, ask the virus to grow in the presence of antibodies, and this is the amount of virus we get after 2 passages. In the absence of antibody, the virus is growing as it normally would to titers of approaching 10 to the 8. If we do that in the presence of single antibodies, the virus has no trouble escaping those individual antibodies and is growing to similar titers. But if we use combinations of antibodies that have non-overlapping resistance profiles, we completely crush replication, and then after 2 passages, there's no detectable viruses. Essentially, we've come at the virus from too many directions for the virus to simultaneously get resistant to both antibodies at once. So that cocktails of antibodies is probably the solution to any potential problems in terms of virus resistance. However, we still have to bear in mind that the monoclonal antibodies that we are using or hoping to use clinically ultimately do come from a population of antibodies that exists in nature, that are naturally mounted by infected individuals. And so it behoves us to actually understand the breadth of antibodies that really are present in convalescent individuals or vaccinees. Basically, how polyclonal and how variable is the antibody response in people that have been infected or vaccinated, because that also will affect the propensity of the virus or the ability of the virus to acquire resistance, not only to these antibodies, but also to the antibodies that are planned to be used therapeutically. So to try to get at that question, we've done essentially the same experiment, used the same approach, a diverse population of spike variants in a recombinant virus. But instead of using a monoclonal antibody to select resistance, what we have instead used is plasma from convalescent individuals. And so we incubate virus with an amount of plasma that's sufficient to almost completely neutralize virus. And then any virus that isn't neutralized, we passage. Sometimes we passage it 3 times, sometimes we passage it 4 times, and then at various points sequence, isolate mutants and ask what selective pressures has plasma placed on virus. And we've done this now with about 25 different convalescent individuals. And the answer is that the outcome is quite variable. So some individuals, like this one here, their plasma behaves almost like a solution of a monoclonal antibody. Clearly, it's selecting for mutations at a particular point in the RBD and does so very rapidly after just 2 passages. Other plasmas, this one here, we have huge difficulty in isolating any resistant mutants. Even if we continually passage the virus in the presence of this plasma, we're not seeing specific variants coming up at any position in the spike protein, suggesting that this is sort of a polyclonal response that's quite well balanced across the spike protein. Others, we have an intermediate phenotype. So this person, for example, you can see variants beginning to come at passage 2, but you really need to grow the virus for longer to see high frequencies of variants at this position. I'd note also that this is one of the individuals from which we isolated a very potent RBD-specific monoclonal antibody. But it's clear that the plasma from that individual has other antibodies that are selecting at a different place in the N-terminal domain of the spike protein. So clearly more than one specificity in that individual. So as I say, we have done this now for 25 or so individuals. This is essentially the entire data set represented on one graph. These points here are places in the spike protein where plasma samples have apparently selected mutations. We can't yet be sure that these are all antibody escape mutations, but I'll show you a couple of examples in a second. But they do seem to cluster at least many of them seem to cluster in what we are now beginning to think of as potential neutralizing epitopes. And the key point here is that it's not one. There clearly are multiple places on the spike protein to which the antibodies can attach and at least impose selective pressure on the virus. As I say, we haven't followed up on all of these variants just yet. That is in progress, but we have some data on some of those variants that is quite interesting. So here, for example, are a set of mutations that were selected using plasma from this individual. This plasma clearly selects for mutations at around amino acid position 150 in the spike protein. So these are the wild type viruses, these are the mutant viruses. And then if we do neutralization assays using plasma from that individual, you can see that the variants that were selected have about an 8 to 10-fold resistance to that plasma compared to the wild type virus. Now, if we use plasmas from different individuals, you see either marginal or no resistance to plasmas from other individuals, suggesting that the dominant antibody response in this individual is different to that in those two individuals. Conversely, if we select mutants using this plasma from this individual, that plasma clearly selected viruses at a different place around amino acid 444, 44, 45. That's in the RBD, and all those mutant viruses are sort of 10 to 50-fold resistant to plasma from this individual. But again, those mutants retain near full sensitivity to plasmas from these two individuals. Again, underscoring the fact that the antibody response in different individuals is at least to some degree heterogeneous. Okay, that's quite important and quite interesting in terms of thinking about resistance. So this slide summarizes essentially everything we know about antibody resistance in the spike protein. So on the right here, what I've shown here are positions where resistance mutations are acquired upon selection with these antibodies, the Regeneron antibodies, or the 25 plasmas that we have tested. And you can see there's some heterogeneity, but clearly some overlap. So this Regeneron antibody, for example, clearly selects resistance that some of our plasmas also select resistance as. Similarly here and here, here and here. Okay, so there's clearly some overlap in the therapeutic monoclonal antibodies and the selection pressure imposed by plasma. Sort of broadening this out to the entire spike protein, everything that's colored in red here is a variant that seems to have been selected by one or more plasmas in the set of 25 that we've analyzed. But the sort of take-home message is that there is some degree of homogeneity, but also a significant degree of heterogeneity, both within an individual and between individuals in terms of the antibody response. And that's actually good news in terms of thinking about the routes of attack that vaccines and antibodies might have against this virus. Okay, let me summarize what we both said. So as Theodora said, convalescent SARS-CoV-2 neutralizing antibody titers can vary very dramatically between individuals. Most have rather low levels, a few have high levels. Some of the high throughput clinical assays are actually quite good at predicting neutralizing titers. Those titers decline, at least during the first 3 months. The half-life of neutralizing titers is about a little over a month. Okay, so they are quite sharply declining, at least in the early periods of convalescence. We can get potent neutralizing monoclonal antibodies from convalescence, and they're protective, at least in animal models. There are rare antibody escape variants in natural populations that we shouldn't be too worried about at the moment, but we will need to keep an eye on. And importantly, the neutralizing antibody response to SARS-CoV-2 is polyclonal and quite variable in specificity. So that's all we wanted to tell you scientifically. We'll just part by making what's a key point. For the last few months, we've really been doing science in a different way. While we ourselves have worked together for many years, over the last few months, we've been part of a much larger group. Importantly, at Rockefeller, Michel Nussensweg and his team, Charlie Rice and his team, we've been basically working almost as one group for the last several months. We've been collaborating with investigators at Caltech, Pamela Bjorkman, at UNC, the animal model work that Theodora told you about. We've also had lots of collaborations with clinical investigators, particularly at New York Blood Center and in the NHS. And I'd just like to highlight these three individuals who, when New York shut down, these guys went to work essentially 24 hours a day to enable much of what we have told you about. Frauke, Fabian, and Jiska really stepped up when things became very serious here. Also, other members of the lab have also worked very hard on this. And so with that, I will stop and invite questions. Thank you very much for listening.
Paul and Theodora, that was really superb. And I want to congratulate you on your work, and also the acknowledgement that there are positive aspects of SARS-CoV-2, and it's brought us together as a community, and it's made us realize that we're all dependent on each other and each other's contributions, and we'll get through this together. There are a lot of questions that people have. So let me start first with the question about the commercial versus the commercial antibody test versus the neutralizing antibody test. Did it make a difference in terms of when these samples were collected over time? Because I'm sure that there were different phases of the pandemic in which the samples were collected.
You mean in terms of correlation, or?
Yes,
so it's very
correlation. Did you see an increase in the neutralizing, the positive neutralizing antibody later on in the pandemic after?
No, we never saw an increase. It's always the neutralizing antibodies always decline, and that seems to be what other groups are finding too. Mm-hmm. The antibodies as measured by the commercial assays do not. And the best correlation between those commercial clinical assays and the neutralization, at least with some of them, are with samples obtained early post-infection. The longer you wait, the bigger the discordance between neutralizing antibodies and antibodies as measured by these high throughput assays.
Yeah. So those samples were all collected sort of within about 3 months of a PCR test. We're now getting patients back 6 months later. It's clear that those declines in neutralizing titer have been maintained. And we don't know for sure yet, but we're expecting that correlation between serological and neutralizing titers to continue to deteriorate. Yes. So it has some implications for convalescent plasma therapy, I think.
So this is a two-part question, but also related to the development of neutralizing antibody post-infection. One, were you able to study children, and were children different than adults in terms of development of this? And did the infection in individuals and the severity of the infection in individuals correlate in any way with the ability to develop a neutralizing antibody response?
Yes, so the more severe cases seem to develop more neutralizing antibodies. The greater the severity of the disease, generally the greater your antibody response. We haven't, and as I mentioned also, the difference between male and females, that's probably in our mind has to do something with antigen exposure. So the more virus you get, the more antigen exposure, and probably the better the immune response you mount against it. We haven't had the chance to analyze any children because, yes, we stuck with adults so far.
Mm-hmm. So, and you get you get your samples from the blood part from the blood banks. Is that right?
From the New York blood part, we've gotten our samples. So we have had 3 cohorts. One that was specifically recruited at Rockefeller to identify individuals and isolate monoclonal antibodies from. One is part of the New York Blood Center that has a huge collection of convalescent plasma, and they were part of the US strategy of obtaining such samples. And the third is the NHS in Scotland, who was just the hospital that was collecting samples from convalescent individuals. So 3 different sources. And the results we get from all of them are very similar. So there's no geographical or appears to be no geographical differences. As far as we can tell.
So it looked like there was a selective pressure based on the exposure to monoclonal antibody. Was it a selective pressure in terms of mutation? And if so, what's the mutation rate of SARS-CoV-2, and how does that relate to other viruses?
Okay, so the polymerase itself is at least 10-fold, 15-fold less likely to make errors during the process of replication than is typical of, I guess, a range of RNA viruses. Although that number can vary quite dramatically. Now, what that means in terms of the sequence diversity, so the rate of change of the virus, so I am not an expert in phylogenetics and sequence diversity, but my reading of the rate at which the virus is changing as we progress through the pandemic seems to me to be even less than that which would be expected based on the differences encountered in the polymerases. So what does that mean? Does that mean that the virus is not encountering strong selective pressures? It could very well mean that. And so that picture that I put up with the transmission window occurring really before there's a significant antibody response, I pulled that picture out of my I made it up, but it might actually be quite accurate. How much immunological pressure has been put on the virus isn't clear to me, but it doesn't smell like it's a lot, to be frank.
So then as treatments develop and as the virus gets exposed to more selective pressures, we may see emergence, is what you're suggesting. Right.
So this is quite complicated. Okay. So for resistance to be an issue in an individual person, right, the population of viruses in that person has to acquire resistance to whatever therapy is applied, and that could in principle lead to therapeutic failure in that individual. For that resistance to be problematic in the broader sense, in the community sense, the virus not only has to be selected to be resistant by that therapy, but then that person then has to go on and transmit that resistant virus to another individual, and in a sense, seed a whole new epidemic of that resistant virus. And given that the infectious window, as it were, for this virus is, well, for someone like me who's used to working on HIV, is comparatively short, this tends to suggest resistance is clearly going to be much, much less of a problem than it is in HIV. Whether it'll be no problem, that's possible, but certainly not guaranteed.
So since you bring up HIV, let me ask a question that has been asked of me several times that I don't have the answer to, and you may not want to comment on, but tell us why a vaccine is so why we can really conceptualize, develop, and test a vaccine in SARS-CoV-2, but we have such a hard time with HIV.
Right.
Yeah.
That could be a longish answer. Do you want to?
I'll start at part of it is what you just talked about. So the mutational rate of HIV is and the speed is on a different level. Mm-hmm. So it mutates a lot faster, and the polymerase definitely makes a lot more errors, and it mutates a lot faster than this virus. So it will avoid anything that targets it extremely fast.
Yeah. In fact, if you take virus and plasma from one infected individual at one time point, the virus that you get at that time point is already resistant to the antibodies in the contemporaneous plasma. That's just not true with SARS-CoV-2, yes. Right?
So, and so if you ask me, at least, if we're going to get an HIV vaccine, I would be very, very doubtful. But if you ask me if the SARS-CoV-2 vaccines are going to be successful, I'm a lot more optimistic. And that has to do with what I mentioned in the beginning. So even patients whose overall neutralization activity in the plasma is not great, they still have the ability to make these really potent antibodies, which suggests, at least to us, that all you need is just to prime the immune system in the right way, which a vaccine has a much better chance of doing than the natural infection, which is probably the majority of the infection restricted in this anatomic site that's not readily seen by the immune system. The vaccine's probably going to have a better chance at inducing a better immune response and the right kind of antibodies.
There's actually another important point about the antibodies themselves. So the antibodies that we've isolated that are very, very potent, they are very close to germline sequences. They haven't gone through a large process of somatic mutation and maturation, yet they're incredibly potent. To get antibodies of that potency and with any breadth in HIV-1, those antibodies have a huge burden of mutation in most cases. They clearly have to dance with the evolving antigen for very much longer to develop that tight binding that allows for potent neutralization. It appears we already have that in our germline for SARS-CoV-2. So it really should be quite straightforward to elicit potent neutralizing antibodies with a SARS-CoV-2 vaccine.
Mm-hmm. Thank you. That was really very helpful. How does age affect the development of neutralizing antibody?
Well, I think that's somewhat complex because there's probably more than one thing going on. Again, I must confess, I'm not, and I don't think you are, experts on the immune senescence that occurs in elderly individuals. But on the other hand, probably for that reason, elderly individuals likely have this higher antigen exposure that Theodora spoke about, just because there's been more virus replication. And so there are both positive and negative influences on the outcome in terms of neutralizing titer in older people, I would say. So
someone develops a neutralizing antibody post-infection. It goes away after 3 months. What's happened with these few people that have been reinfected? Have you been able to study it? What do you think? If not, what do you think would happen?
So, well, we do have to admit we're a little bit on thin ice and a bit, because the numbers that have been studied are quite small. The first thing I would say, this 3-month number, that's there's huge variation either side of that. There are some people that make hardly any antibodies from the beginning, and others that have titers in 20,000. And so, at 3 months, there's going to be some fraction where the antibodies have fallen below detectable levels. So with reinfection, again, hesitant about making predictions, but if you just look at the range of what happens in the world of viruses, so with HIV, there's probably no such thing as serological immunity. With measles, there's lifelong protection that's probably mediated by serology. Probably the viruses that are going to be the closest mimics in terms of what will happen in terms of reinfection are the seasonal coronaviruses. And there we sort of know there is protection in convalescence, but it wanes over time, and sort of 12, 24 months later, I think it's clear that most people, or at least a large fraction of people, can be reinfected. It's not clear to me why SARS-CoV-2 would be any different to that. Similar virus, similar lifestyle, similar type of neutralizing response, I think.
Let me ask you one final question. What implications does this have in terms of the frequency in which someone would have to be vaccinated?
Yes, we don't know. It depends on the vaccine. And again, as Paul, we can only predict at this point. It is possible that it depends. So what we're hoping for is that the antibody response to a vaccine will be much better than what you get from natural infection. So much higher levels of neutralizing antibodies. Now, how long they will last is a different question. So it might be that you will require boosters once a year, similar to a flu vaccine that's annual. It might be that it lasts for a few years and then you need another one. Maybe some certain platforms can deliver antigens for a long time, so they would persist over time. It's very hard to tell, and there's a lot of different vaccine platforms being developed at the moment. So it's very likely that it will differ depending on what vaccine you get.
Yeah, I guess I'd basically say the same thing in summary by saying that what happens in convalescent immunity to natural infection, I don't think in any way can predict what would happen in the context of vaccination. And, even if we do have to go back every year for a booster, that at this point would be a win.
This has been a great discussion. Thank you so much for your presentation, and thanks so much for the work that you're doing.
Thank you. Our pleasure.